Well, thank you very much for your kind introduction, and thank you also for the invitation for you who were responsible for it to come here. It's for me the second time being in Chengdu, and certainly we're going to enjoy it very much the ride tonight. Now, I'm now turning more to a medical story, and I think I selected a title which may sound a little bit provocative, but I think in the end you will understand it to some degree. Well, I think basically when I received the invitation here by Richard, I was a little bit hesitant in the beginning because I thought if I look back at my own experience in science, is it really serendipity? Was there so much serendipity? Or was there a lot of it which was hypothesis driven initially? And when you see the picture here to the right, I think you can clearly follow Marty Schalke's remark. You start first to wonder. And maybe that leads to some interesting aspects subsequently. Now, when I started to become interested in science, it was a very early stage. In the early post-war period after the Second World War, in Germany at least, no books available, but the first issues which came out were bibliographies of Robert Koch and also Louis Pasteur subsequently, which I was reading at the stage, and I must say it fascinated me from the very beginning. The fact that they made a number of enormously important discoveries at the end of the 19th century, I think was something which was really driving me in the end into studying medicine. Of course, during the study of medicine, I became aware of a large number of human diseases which certainly at that time, but even up to today, are not really clarified as far as etiological aspects are concerned. Even if you think of many of the cancer cases, or cancers, think of neurodegenerative diseases, you think of cardiovascular disease, and a couple of others. There are many aspects which came up during these years, but in a way, there are also a number of questions which remain totally unclear up to today. Now, if I look back on my own professional career, I would say there were four phases where I would rather say the research activities were certainly more hypothesis-driven, but on the other hand, several serendipitous observations came along during this period of time. I would say the very first phase was really led to my interest into the question to which extent contribute infections to human cancer. That's never left me up to today. And in the very early period of time, I thought a very good approach for that might be to look into the question to which extent infections cause chromosomal modifications. Chromosomal modification at that period of time, just the chromosome spreading techniques had been developed. It was a very early phase, which interested me very much. I finished my medical training and went into the Institute of Microbiology in Düsseldorf, which was probably the most boring period of my scientific life, because at that time, the 3 years, I could do whatever I wanted to. There was sufficient money available, and but there was no guidance. And whenever I went to my former boss, proposed something which sometimes was really a little bit ridiculous, he listened carefully and said to me and said, well, it's quite interesting, why don't you try it? And so in a way I got very frustrated during this period. But looking back right now, it helped me, this period of time helped me tremendously. And I would wish for many of the young people right now that they have either during their doctoral period or in the postdoctoral period, a period of time where they can make themselves familiar with a broader range of subjects and interests and look into those questions which might be most interesting for them. So, to make a long story short, I started initially to work on virus-induced chromosomal changes and cancer. Indeed, the work was not totally unsuccessful, at least for specific viruses. Adenovirus type 12, I could identify at that stage some very specific chromosomal modifications occurring under those types of infection, which are listed here in chromosome 1 and human chromosome 17, but it didn't lead from there to any further understanding of events which are linked to human malignant diseases. Subsequently, and I will dwell a little bit more on this in a moment, between the years 1966, so for many of you clearly in prehistorical times, between 66 and 1972, I worked mainly on one of the tumors which was suspected to play a role in human cancers, namely it's an African tumor occurring in children at the age of 5 to 12 years on average, and these tumors were absolutely fatal for those kids. They died usually quite quickly afterwards, but Dennis Burkitt, a British surgeon who had claimed at that stage that the etiology of this tumor shows some very peculiar characteristics. Namely, it occurred in very specific regions in Africa, at very specific altitudes in those regions, and that he postulated, since it was similar to the development, to the malaria falciparum, to tropical malaria, that it might be an insect-borne vector who carries the virus and leads to this type of cancer. And indeed, after a certain period of time, Anthony Epstein, at that time in Bristol, discovered that there is indeed, in some cell cultures obtained from these tumors, you could identify a very, very small percentage of those cells, a virus, the Epstein virus. Of course, the electron microscopic demonstration didn't prove anything in that case, and nobody knew at that stage which virus it is. It was clearly a virus of the herpes virus group, but herpes viruses were known, human herpes simplex viruses, cytomegalovirus, varicella zoster virus, the chickenpox virus, and a couple of others were already identified at that stage. And this agent, the cell lines which produce occasionally in a small percentage these virus particles, came into the laboratory of the Hausen in Philadelphia, and I will come to this a little bit later. There was a third period in 72 to 201 when with my group studied mainly papillomavirus and cervical and other cancers, but I will come to it later on, and certainly up to today we have a chance to work on specific other aspects, again, of infections linked to human cancers, potentially linked to human cancers, but also to certain types of other diseases, which, at least from our scope, turn out to be extremely interesting and exciting. Now, let me come briefly to the period in Philadelphia, after finishing the Düsseldorf period in a lot of frustration, without any real result during this period of time. I came into the laboratory of Philadelphia with a rather clear-cut idea what needs to be done. And the idea was really based on previous experiences or previous reports that certain bacteriophages insert their DNA into host cells, into bacteria, and these bacteria change their properties. And it was a simple and to some degree naive idea. Is it possible that cancer, for instance, in humans is based on the same kind of development, that infections take place, the genetic material is picked up by the respective agent, by the respective bacteria, and subsequently, in this case of cancer, that the cells which picking up new genetic material from infectious agents may change their properties, may develop malignant growth properties. I had indeed at that stage heated arguments with Werner Henle and Brigitte Henle at that stage, because he was convinced that the small number of induced cells which produced virus in the Burkitt lymphoma lines was due to persistent infection. Interferon would play a significant role in keeping the infection at a very low level, but I speculated very much that this might be an agent which is present in every individual cell, and like in lysogeny in bacteria, that there would be a spontaneous induction taking place in some instances. And then we decided firmly to look into this question quite deeply. At that stage, see here in the following picture, at a relatively young age, and here is a technical assistant, Elaine, who was the one who was instrumental for the Henles in finding that infectious mononucleosis is caused by Epstein-Barr virus, and that she developed infectious mononucleosis during her period at the Henles. They had serum taken prior to her disease, and also subsequently after they developed the disease, and it was clear indication that sky-high antibodies developed against the agent which was found in Burkitt lymphoma cells, and then a subsequent study very elegantly and clearly showed that the virus is indeed that infectious mononucleosis is always going along with a high antibody titer against the Epstein-Barr virus in these cases. Well, during the period of time in Philadelphia, then it's permitted me for quite a while to continue to work on virus-induced chromosomal changes, but urged me to start early to work also on Burkitt's lymphoma cells. And one of the urgent questions for the group in Philadelphia was initially that they could discover in Burkitt's lymphoma cells, with many of the human sera, almost a large percentage of human sera, antibodies against the low number of cells, and of course it is suspected that these were the ones which produce the Epstein-Barr virus, and indeed they asked me whether I could test the individual fluorescent cells for those types of, whether they contain the virus, which was a little bit laborious, but it turned out to be successful indeed. And I picked up individual cells and had them sectioned for electron microscopy, and indeed it was possible to demonstrate that the virus was present in every cell. There were a couple of interesting and more serendipitous observations made during this period of time, and one of them was what we called at that stage, sorry, I'm going too fast. What we called at that stage the incomplete chromosomal condensation, and indeed it turned out that very specific chromosomes, particularly in leukemic lines, very specific chromosomes showed some uncoiling, and at that stage I didn't understand really why this is happening, and it was always involving more or less the same chromosomes, in particular chromosome 1, but also in many instances chromosome 17 and 16, and basically that was quite for me an interesting observation at that stage, that this was a delayed replication of DNA, which was taking place here in these uncoiled structures during the mitosis. DNA synthesis was still going on in these types of structures. So in a way, this was not intended to work on, but this was clearly serendipitous. In addition, during this period of time, I was finding Stockholm at the Wistar Institute, we could demonstrate that adenovirus, which is immortalizing hamster cells, which is even causing adenovirus type 12, which is even causing malignant tumors in newborn mice and in newborn hamsters, that the DNA under those circumstances is integrating into the host cell DNA, but not as a total genome, but only in specific fragments. Well, so in a way, the period on working on chromosomal modifications did not work out very well, but on the other hand, the period on Epstein-Barr virus turned out to be quite successful in the end. Because indeed it was subsequently possible to demonstrate, after I moved to the Institute of Virology in Würzburg in Germany, to demonstrate that indeed every Burkitt lymphoma cell, irrespective whether it was virus producing or not producing, did contain viral DNA by in situ hybridization, newly developed method at that period of time, and also by demonstration of the viral nucleic acid in virtually every DNA in every biopsy from Burkitt lymphomas, and also from a different type of tumor, nasopharyngeal carcinoma, and even here the early in situ hybridization was still a little bit messy. Subsequently in Hong Kong, they were much nicer done by Dr. Loh and his colleagues, but we could clearly demonstrate that the epithelial cells in nasopharyngeal carcinomas did contain by in situ hybridizations did contain the Epstein-Barr DNA, and this settled a long-standing question, in particularly among immunologists, who claimed that the nasopharyngeal carcinoma, which is a tumor where the epithelial tumor cells are surrounded, embedded virtually in a large number of lymphatic cells, and Epstein-Barr virus at that stage was already quite clear to be a lymphotropic virus, that the viral DNA should be in the lymphatic cells, infiltrating lymphatic cells, and not in the Burkitt lymphoma cells. Not in the cancer cells, epithelial cancer cells. Clearly, that was possible to demonstrate it quite well, and this was later on confirmed in quite a number of additional laboratories. Well, subsequently, I moved again to another place in Germany. At that stage, I started to work on a very different tumor, namely cervical cancer. Why cervical cancer? Because cervical cancer was basically the prime candidate for being involved in being linked to viral or to an infectious etiology. Already in 1842, an Italian physician had noticed that it must have something to do studying the mortality statistics in Trento, that it must have something to do with sexual contacts. And subsequently, a large number of studies have been conducted for quite a number of years on trying to link sexually transmitted diseases, in particular gonorrhea, syphilis, lambliosis, and trichomoniasis, to this type of condition. The data were clearly not convincing, and it was only at the end of the 1960s that another candidate popped up, namely human herpes simplex virus type 2, the genital type of herpes simplex virus, producing relatively nasty lesions in the genital tract of those people who are affected, which are recurrent. And this was mainly based at that stage on serological studies, because many of those women with cervical cancer had elevated antibodies to human herpes virus type 2, and so it was quite natural to look for the DNA of human herpes virus type 2 in genital cancer biopsies. In fact, we had initially done it with Epstein-Barr virus, which was a very difficult system due to the low number of virus-producing cells and due to the fact that the virus could not be replicated and propagated in any type of cell culture. So I thought it must be very easy to do the same with human herpes virus type 2. We tested close to 100 individual biopsies for human herpes virus type 2 DNA and failed entirely to find in anyone any evidence for herpes simplex type 2 DNA, quite in contrast to the Epstein-Barr virus data which we had obtained previously. So it was, for me at least, quite clear that the virus most likely does not play a role, and in fact I presented these data at a meeting here in the States, in Florida, and which was devoted to the herpes type 2 cause of cervical cancer. And this was for me the most unpleasant experience I ever had in my scientific life, because everyone was telling us that our methodology was not sufficient, particularly because another colleague here from Chicago claimed at that stage that he had found about 40% of the human herpes virus type 2 genome in one single biopsy of cervical cancer. Well, in a way, nobody, of course, trusted the data. But even before this event, I had a chance to review the available data on genital warts and cancer. And that was quite interesting because looking back for almost 100 years, it was possible to find about almost exactly something like 100 reports, anecdotal reports, that genital warts occasionally convert into malignant tumor. Since I had seen myself that genital warts, by looking into them by electron microscopy, contained typical papillomavirus particles, which was known, of course, at that stage already. Suspecting that this agent, which is present in genital warts, particularly after infection of cervical sites, might have some different properties and lead to cancer in those sites, which might be much more susceptible for malignant transformation than the usually external genital sites. So initially, since there was no virus, not a lot of virus being found in genital warts, the methodology was not really very much advanced during this period of time. I started to grind up plantar warts just by testing with the mortar and sea sand. I can assure you it was a very successful period, and a very successful technique, because we could purify very easily, and subsequently by density gradient centrifugation, large quantities of papillomavirus from genital warts, and that gave us the first hint, by labeling it in vitro radioactively, that this agent, which we isolate from plantar warts, had probably nothing to do with genital warts, and that the agent in genital warts must be different because none of the genital warts biopsies which we analyzed ever showed any signs of hybridization with the plantar wart virus, and this accounted also for a large number of cutaneous other wart lesions. So from that moment on, it was quite clear that the human wart virus, as it was claimed at that stage, didn't exist. But these are a multitude of viruses, and up to today, far more than 200 individual types have been isolated and characterized, which clearly shows that there's a wide variety of these agents available. Now, so initially we started to work on those agents to characterize in particular the plantar wart viruses, but of course the most interesting question was, what is the nature of the genital wart virus, and does this genital wart virus have anything to do with the development of cervical cancer? And it took us quite a while before we were able to isolate sufficient quantities of papillomavirus from the genital wart to clone this virus. Also, the cloning procedures were still in a relatively early stage at that period of time, and that we could, I postulated it earlier in the paper, that we could then identify this agent and look at the cervical cancer where we would find it under those circumstances. The data were amazingly frustrating because we didn't find it in cervical cancer biopsies. But by using this type of probe, it became possible to isolate a closely related agent from a laryngeal papilloma. And this agent, now labeled as HPV11, was clearly a much better candidate in comparison to the previous one. We found in so-called southern blot hybridization some faint bands by annealing the HPV11 probes with the DNA from cervical cancer biopsies. First of all, I should mention, as I showed you, we cloned the HPV6 DNA initially. Lutz Giesmann, the HPV11 DNA, very talented co-workers, and she's my wife as well. And Matthias Durst subsequently cloned one of these faint bands after many, many frustrating attempts, he isolated about 250 clones which were all negative, but the 251st clone turned out to be a hit because it turned out what we later on labeled as HPV-16, and Michael Bossart at that stage, a medical student, he was able to clone another agent, HPV-18, and both of them turned out to be extremely interesting because from the very first moment it became clear that 50% of cervical cancers biopsies were positive for HPV-16 DNA, and even slightly less than 50% and slightly more than 20% for HPV-18 DNA. And in fact, if you look at it globally right now, it's just the opposite. Slightly more than 50%, 16% and slightly less than 30% for HPV-18. But this changed from the beginning quite substantially. A couple of other isolates have subsequently been obtained, some of them in our laboratory. This is this labeling here because it should be HPV-30, which was isolated in our lab as well, but also Gerard and a couple of other groups were able to identify some others, which by now bring up the number of positive results to about close to 100% of cervical cancer biopsies. Virtually everyone is positive for any of these types of agents. And indeed, the vaccination story has been mentioned already. It was a very frustrating story in the beginning because attempts to persuade the pharmaceutical industry at that stage, I tried it initially in Germany and in Switzerland, to produce a vaccine against these agents were very frustrating, except for one company in Marburg, which was the Behring company. The director was quite interested, but since it belonged to another big pharmaceutical company in Germany, one of the directors asked for a market analysis, and the market analysis turned out to be absolutely negative. And in addition, of course, they claimed that everyone already as a baby is infected by the same agent. Everyone has antibodies, so it wouldn't make any sense to develop this type of vaccine, which left us in some frustration until subsequently 2 US companies initially started it up in the end of the 1980s to work on these types of vaccines. Anyway, it brought some delay in the development of vaccines and left our group at least for a certain while in some frustration. But anyway, a couple of other interesting experiments came up during this period of time. I didn't mention it that in Burkitt's lymphoma, two events seem to be, at least in the endemic form of Burkitt's lymphoma, mandatory. One is the uptake and persistence of Epstein-Barr's DNA and also its specific genetic expression. And secondly, also the rearrangement of the c-Myc gene, which becomes linked to promoters of the immunoglobulin at chromosome 8 and chromosome 22 in most of these instances. And it was an interesting question for us to look into the fact what happens if we fuse from the same patient who has a Burkitt's lymphoma, these cells will lymphoblastoid cells, again obtained from the same patient. To which extent does it affect the c-Myc expression? Interestingly, it does not affect the c-Myc expression, showing us at that stage that c-Myc expression by itself cannot be sufficient for Burkitt's lymphoma development. And secondly, one other interesting aspect was here that the fused cells, if inoculated into incompetent animals, they can use mice, these cells initially form tumors for about 2 weeks, 3 weeks, and then a central necrosis develops, and it's really like, it was previously mentioned, like a wave. One part was really an apoptotic debris which was left from the necrotic regions here in these animals, whereas in the other part still mitotic events were going on for a short period of time until all of these cells died and the lesion completely dissolved under those circumstances. Well, this was an interesting observation, which also was followed by another observation, which was to some degree serendipitous, namely that we could demonstrate that in vitro immortalized cells, human cells, by specific genes of the, for instance, human papillomavirus type 16, the so-called E6 and E7 genes, which are uniformly expressed in HPV positive tumors, that those types of cells are non-malignant if inoculated into a human mouse. But in addition, if you keep them for prolonged periods of time in tissue culture, they gradually turn over to become malignant lines. That was the first indication for us that even the viral gene expression by itself is obviously not sufficient for the tumorigenicity. But there was one other aspect which we investigated in this stage, namely to look whether in those animals here, like excuse me, like in the like in these nude mice, but under those circumstances, the immortalized lines, which are not yet malignant, whether they express the viral genes to the same extent as later on malignant cells. In vitro, they are virtually indistinguishable in tissue culture. But if inoculated into animals, it turned out that within 3 days, the transcription rate of those viral oncogenes is being turned off, switched, or virtually completely switched off. They form little nodules which persist but do not continue to grow subsequently. Whereas in malignant lines, the transcription goes on from the very first day, continuously in those circumstances. But there were a few other things which came up which were at this stage really serendipitous. Namely, I noted at that stage that specific tumor promoters, the 4-ball esters, particularly the so-called TPA 4-ball ester, which is a very potent promoter, tumor promoter, that modified the structure of Burkitt's lymphoma cells which produce EBV, and it turned out that they were indeed extremely induced, so that you get more than 50% of the cells in part producing suddenly the virus, and this is even by now a very convenient method in obtaining large quantities of the virus by accident, really, by studying African green monkey cells, which lymphoblastic lines, in order to see whether they contain related Epstein-Barr-related virus. Indeed, we found this virus in African green monkeys, but we noted that there was another virus present in those types of cells, which we could identify, as we called it initially, a B-lymphotropic polyoma type virus. We saw instantly that human sera, at least 20% of human sera, contain neutralizing antibodies to this monkey virus, and we tried at that stage desperately to find an equivalent in humans, which we didn't find. Indeed, by now 2 groups in Denmark and Italy have been able to identify that agent. It's very closely related, of course, as can be anticipated, to the green monkey isolate, and it's now called human polyomavirus type 9. Another observation which we made during this period of time was the identification of human adeno-associated virus type 5, which turned out to be a rather interesting vector system at this stage for a number of experimental approaches, because it has a high affinity to cells of the bronchial tract. And it came along because I tried at that stage to infect human water material with adenovirus, with an adenovirus, in order to see whether there would be a helper effect on the suspected wart viruses in these cells. But what came out was really this picture here, namely a huge amount of adeno-associated viruses, clearly distinguishable from the morphology, an inhibition of the adenovirus replication and an enormous amplification of those other agents. This observation turned out to be, or is turning out even right now to be quite, in our view, quite important. Also it was at that stage clearly serendipitous, namely that we saw subsequently that a large number of adenoviruses, of herpes viruses, even vaccinia viruses, are able to amplify a co-infection of a cell which harbors other small DNA agents, to amplify these agents to a very large degree. This accounts, for instance, for all kinds of polyomaviruses which have been tested. It accounts for papillomaviruses even which have been tested. And it's also, as we see, that I will very briefly develop this at the end of my talk, also for other single-stranded DNA agents. It seems even that human TT viruses, as our group could identify, in Epstein-Barr producing cells produce particles which are most likely those types of TT viruses which are very difficult to demonstrate under those circumstances. So here are some of the herpes viruses which have been tested producing these agents. So the reactivation leads to the amplification of the small viral genomes, and virtually all these instances to an inhibition of the replication of the reactivated herpes viruses, which seems to account for all of these agents under those circumstances. In fact, this leads me to a number of serendipitous observations which have not been resolved until today. Namely, first of all, we do find a number of very peculiar particle structures in cells which have been, leukemic cells in this case, infected by human herpes virus type 6, which show a rather strong affinity to the envelope of the human herpes virus type 6 envelope. So far, they have not been characterized fully. It's an interesting observation that still requires further studies. If we infect a specific line of Hodgkin cells, kept in tissue culture, with herpes viruses, in this case the human herpes virus type 1, a number of rather remarkably interesting, very homogeneous types of particles show up under those circumstances. Again, an open question which needs to be resolved and which has not been resolved. Interestingly, you find such types of particles, at least which are morphologically very reminiscent, in the so-called hairy leukoplakias in AIDS patients, which under those circumstances, and Greenspan initially demonstrated in San Francisco, which very much morphologically resembled these particles in the presence of cells which produce large quantities of Epstein-Barr virus at the same stage. And one treatment of specific rapid tumor cell lines with tumor promoters, again, similar particles show up under those circumstances. Again, not identified until today. It's not only identified by our groups, these were data from our own group here, but it has also been demonstrated by other groups here already in the 1970s, and still the question remains unresolved about the nature of these particles. Interestingly, the two other types of agents, which are both oncogenic at the same stage, namely, it's blocked somehow. Doesn't move. Two other types of agents which are oncogenic, namely the Marek virus of chicken and also the UK frog virus, both due to herpes virus infection. Here the herpes virus particles produce similar particles, and as I said, frequently observed, but in their nature up to today not clarified. This is a curious observation in a melanoma cell line of similar particles here at the high magnification, which have also not identified. And here another one of Hodgkin line, again, in fact, simplex files showing a number of these particles. Indeed, we also had some unpleasant experiences because we saw some particles different in size and shape, of course, in human prostate cancer lines. They turned out to be murine leukemia viruses. These lines had been presaged in nude mice and acquired the endogenous retroviruses, replication competent endogenous retroviruses, which had been transmitted to the human cells. I think the next one will skip. There was a couple of pleasant events which came up subsequently, which I'm showing here in Stockholm, and these are my three sons at the same time who were present in Stockholm, and even a sketch which my sister-in-law did during the Stockholm meeting, and obviously yes, she was a little bit bored apparently by the ceremonial aspect there. Now let me finish by just coming to the last period. So up to here I would say the first basically hypothesis-driven approach to chromosomal breaks linked to virus infections, and hopefully what I expected at least at that time to be linked to cancer didn't work out very well. Clearly the Burkitt lymphoma story worked out quite well because we could indeed demonstrate that every tumor cell contains viral DNA which is genetically active. So the papillomavirus story worked out quite well. But we came across in recent years, I've talked on other occasions about this many times, I will be very, very brief, that indeed it's an interesting observation that we do know a number of human agents here which are non-tumorigenic in humans, but upon transmission to other species are highly oncogenic under those circumstances. And this raised the suspicion this is something similar in our domestic animals, which upon transmission, which does not cause tumors in these animals, but upon transmission to species in which they cannot replicate, so those viruses do not replicate here, that under those circumstances they might become tumorigenic. And the basic question behind it was, in the end, is it possible that we, during the past decades, overlooked or ignored the risks of zoonotic latent infection with long latency periods, often for more than 30 years. We do know at this moment latency periods for some of the infections up to 60 years for specific cancer and other chronic infections. And indeed, this led to a careful study in particular in cattle populations, and we could correlate the incidence of particularly colon and to some degree also, as we will hear in a second, breast cancer to regions in which particularly specific breeds of cattle are being prevalent, namely our European, Asian type of dairy cattle, which is in all these regions where those cattle populations are being kept. These populations are quite frequent, and in addition, colon cancer and breast cancer share to some degree the same epidemiology, and some regions which are quite remarkably different and inconsistent in that respect, because they show, for instance, like Mongolia, a low rate of colon cancer and also a low rate of breast cancer. As I'm sure most of you know, red meat has been linked to colon cancer and red meat consumption, but it's remarkable that Mongolia is one of the countries with the highest rate of red meat consumption, and yet the colon cancer rate is very low. In Bolivia, the situation is somewhat analogous here. It's a low rate for colon cancer, slightly higher rate for breast cancer, and India is, for other reasons, a very remarkable other example. But to make a long story here short, we have isolated a larger number of specific small molecules apart from serum of dairy cattle, in part from milk of dairy cattle, and many of these agents here, particularly in this group, have been identified in milk, dairy preparations for our European cattle, and interestingly, two of them have been isolated from lesions of multiple sclerosis. They're quite similar to agents which a group in Yale has isolated. Laura Manuelidis, at least one of them is quite similar, from spongiform transmissible encephalopathies. And she claims that this is potentially an agent which may be linked to the etiology of those types of diseases. So this led our interest really also into neurodegenerative diseases, in particular the fact that we could isolate them here from MS brains. In a way, it led to models which I think I will not discuss intensively right now, how we foresee at least an involvement of those agents in this type of cancer. I will talk on other occasions more intensively about it, namely that we pick up those types of agents due to the consumption of raw undercooked red meat, but we still do not know whether well-cooked red meat does not contain infectious agents under those circumstances. It's a consumption of what has been described in the past of cured and fried or barbecued red meat leads to the formation of chemical carcinogens. I would have here to mention that we do know right now that there's no viral or infectious etiology of any cancer which does not lead, in addition, mutational events occurring probably either as genetic events or as epigenetic events within this very same cell prior to the malignant conversion. So this was the basis of the speculation. Inherited genetic modifications would basically play the same role. For milk and dairy products, the situation would be slightly different because of those circumstances. There would be an early uptake, probably during the weaning period of new babies, of those types of agents, and indeed there's good evidence that we all get infected by those agents, and we do know right now that they can infect human cells, or at least they are genetically active in human cells, and so in a way we need to clarify the situation, whether they play a role or not. There's some evidence linking at least multiple sclerosis to these types of infections, but the data are still a little bit premature in order to talk in detail about it. So basically, that's the thing for young people right now. During my period when I worked on HPV, we were frequently ridiculed of working on a virus for which everyone knows that it's absolutely harmless and doesn't cause any tumors. So quite a number of peers I would say not rather remarkably fair in their comments. Today we experience to a lesser degree something similar by postulating that milk and dairy products and serum on red meat plays a similar role. So even if you can face your fears, defend your good ideas, and as I say, last slide I'm showing, occasionally some of them might be very aggressive, so don't be afraid and stand up. Thank you very much.